We've looked at the diversity of plants and some protists and how those things have evolved. Now let's look at animals. This is going to be an animal diversity blitz. We're going to go through a lot of different genera of animals and introduce you to their different taxonomic groups. And while doing that, we're going to lead into a conversation about ecology and about how animals interact with each other, animals interact with plants, and how these systems form communities and how those communities function on a larger scale.
We'll start with this family tree. You can see that the ancestral animal is a colonial protist. This protist was likely very similar to some of those protists we looked at in earlier lectures, possibly flagellated on a stalk, but organized in a colony, maybe something like volvox, except heterotrophic. The first group of organisms that is an outgroup from the rest of animals are the sponges. Sponges don't have true tissues. All the rest of the animals do have true tissues. Sponges are filter feeders, and they're essentially a colony of cells that work together as a functioning organism. Pores in the structure of sponges let water in and out, and flagellated cells, called kyanocytes, bring in material to be digested by amoebocytes. They feature a skeleton made of strong fibers. Maybe you felt a natural sponge and used one in your home to clean things with. Well, that's the leftover skeletal fibers of a sponge. Sponges occur in marine and some freshwater habitats all over the world. What separates sponges from the rest of the animals is the fact that they don't have true tissues. The group of animals that have true tissues can be split into two groups. One is the group that has radial symmetry, and the other is the group that has bilateral symmetry. On the far right, you can see an example of the sponge. These organisms are asymmetrical. If you tried to make a copy of one side from the image of the other, you couldn't do it. An asymmetrical body plan does not have a mirror image of itself, no matter how you look at it. In an organism with radial symmetry, there are many different points where you could double the image of the body plan and it would create a full image of it. In this example, you can see that if you put a mirror on any one of these planes, you would essentially replicate the full organism. In an organism with bilateral symmetry, there's only one plane that creates a mirror image in a complete organism. So let's look at some organisms that have radial symmetry. And the first group are the cnidarians. Cnidarians have several different body forms. One is the polyp form. In a polyp form, the cnidarian is grounded or rooted onto the substrate. Examples of polyp cnidarians are the corals, sea anemones, and the hydra. You can see that the digestive system of cnidarians is simple. There's a mouth that's also the anus. There's also a gastrovascular cavity where digestion occurs. The tentacles are often used to capture prey, which is then moved into the mouth. In the medusa form, cnidarians are free swimming. You might be familiar with jellyfish. Jellyfish also have a single gastrovascular cavity and also a single opening that serves as both the mouth and the anus. An important feature of cnidarians is the presence of cnidocytes. Cnidocytes are specialized cells that sting and capture prey. Oftentimes they envenomate prey and paralyze or kill them. Cnidocyte stinging cells occur along the tentacles of cnidarians. They're ready to fire upon contact with the prey item. The cnidarian body structure uses what's known as a hydrostatic skeleton. Its form is maintained by internal pressure. The polyp form of cnidarians includes a stalk that holds its tentacles upwards, while the medusa stage is free living and often like a bell. You can see on the bottom left an example of a moon jelly. In the center is a sea anemone, something that you might see in the local tide pools or maybe if you're scuba diving in the water. On the far right is a hydra. Hydra are cnidarians that occur in fresh water. One important thing to note at this point is that all of these examples have radial symmetry. As long as you split these body forms along the center axis and double it, you'll make the complete version of the organism. Now let's look at some organisms that have bilateral symmetry. The organisms with bilateral symmetry can be split into two groups, the protostomes and the deuterostomes. The protostomes include flatworms, rotifers, mollusks, annelid worms, roundworms, and arthropods. The deuterostomes include the echinoderms and the chordates. We talked a little bit about animal development, but let's review it again. Remember that in the sea star embryo, we saw gastrulation and the creation of the gastrula. The cells in the gastrula stage are beginning to differentiate. The ectoderm layer becomes the skin and the central nervous system. The mesoderm layer becomes organs and muscles. The endoderm layer becomes digestive lining and digestive tract. The blastopore, which is the opening that leads to the archenteron, becomes the anal opening in deuterostomes. In protostomes, it becomes the mouth. This is an important distinction between the deuterostomes and the protostomes. Let's look at a few taxonomic groups in order to see the different kinds of body plans that bilateral organisms have. We'll look at three groups: the flatworms, the annelids, and the roundworms. Remember, all three of these groups are protostomes. Here's an annelid worm. You might be familiar with annelid worms. Those are like the earthworms you find in your garden. Let's look at a cross-section to see what the tissue layers look like. You can see that the very outside tissue layer is ectoderm. Within that is a tissue-filled region from the mesoderm. Within that tissue-filled region, there's a space. The presence of that cavity, called a coelom, is important, and we call this body plan a coelomate. Within an inner layer of mesoderm, we find the digestive tract. Take note, the digestive tract is surrounded by endoderm. Here's another body plan, illustrated by the nematodes. This is a pseudocoelomate. Again, we have a body covering a tissue called ectoderm. Within that, we have mesoderm. But now we have a digestive tract that is not surrounded by endoderm. Again, we have a space, but since this space is not surrounded by endoderm, we call it a pseudocoelom. We call these kinds of organisms pseudocoelomates. Nematodes are pseudocoelomates. This species of nematode is called Dracunculus medinensis. Dracunculus is also known as the guinea worm. Guinea worms cause infections wherever they are present in the water. They first infect copepods in the water. If the copepods are then ingested by humans, the larvae develop into mature adults that eventually exit the body. These mature adults can be as long as a meter. Several communities have worked with philanthropists, scientists, and doctors to almost completely eradicate the guinea worm infection from humans. The last body plan we'll discuss are the acoelomates, that is, organisms that lack a coelom. To illustrate this, we'll look at a flatworm. You can see that the flatworm is on the far left of the taxonomic tree. On the bottom right, you can see that the flatworm has ectoderm, mesoderm, and a digestive tract that stems from the endoderm. The acoelomate does not have a coelom. Now that we've discussed these three groups, let's move on to another group of protostomes, the mollusks. While we don't know everything about the evolution of mollusks, we do have a good idea of what an ancestral mollusk looked like. This is the archetype mollusk. It's a proposed organism that contains all of the features that mollusks share. For the purposes of this class, we'll discuss a few important features. The first is the radula. The radula is a rasping organ. It's used to feed. You can see here in this cutaway of a snail, the radula is used to scrape off organic material from the substrate. Here you can see that the radula has a lot of different teeth on it. It's a coarse scraping tool. The archetype mollusk also has a mineralized dorsal dome-like shell. Underneath this shell is a soft bed of tissue called the mantle. Some mollusks have lost their shells over evolutionary time, but maintain their mantle. Mollusks also have ctenidia. Ctenidia are breathing organs, like gills, that must remain wet to function. The first group of mollusks that we'll talk about are the bivalves. The shells of bivalves are hinged with interdigitating teeth. You can see that on the top right. If you had the other shell, those teeth would groove and lock in together. The ancestral foot of the mollusk is now used as a blade for digging. Some bivalves use their ctenidia for filter feeding. Some bivalves can also swim. Locomotion is accomplished by bivalves when they open and close their shells rapidly. This forces water in one direction like a jet. Doing this, they can shoot across the sea floor. The next group of mollusks are the gastropods. The gastropods are a highly diverse group. Earlier we talked about the cnidarians. This is an example of a cnidarian, Physalia physalis. This is also known as a Portuguese man-of-war and is a very dangerous cnidarian. On the bottom right are the stinging cells, the nematocysts. You don't want to get stung by the Portuguese man-of-war. Here's a gastropod that feeds on the Portuguese man-of-war. It's called Glaucus atlanticus. This marine gastropod floats along and eats Physalia. It also eats other organisms, but when it eats Physalia, it takes in the nematocysts and uses them as their own. So if you pick up a Glaucus atlanticus, you could get stung. That sting did not come from the Glaucus, it used it from the Physalia, part of its diet. Another interesting thing about Glaucus is that the bottom side is silver. This is so that if predators are looking at Glaucus from below, it matches the silvery sheen of the sea surface. When looking down at Glaucus from above, its dark colors help camouflage it against the dark sea floor. This is called countershading, and it's an evolutionary strategy that we're going to see more of. An important subclass of gastropoda is Pulmonata. These are the terrestrial and freshwater gastropods. These include snails and slugs. This is a familiar species that you might see if you're anywhere along the California coast. Areolimax californicus, the banana slug. On the right you can see that the mantle of the slug is clearly visible, but it lacks the shell on top. Remember that the mantle is a soft bed of tissue below the shell. Slugs have lost that shell over evolutionary time. The next group of mollusks we'll discuss are the cephalopods. The shell in cephalopods, if present, is often reduced. On the three examples of cephalopods on the top right, you can see the far right example is called a nautilus. This cephalopod has a shell, and is thought to represent a more ancestral form of the cephalopods. Zoologists believe that a snail-like marine ancestor, like the nautilus on the top right, evolved an ability to adjust gas levels in their shells. That made their shells more buoyant, and eventually gave them the ability to float or swim. This opened up a new dimension of niches for evolution to take its course. In essence, an ancestral snail-like mollusk in the ocean was able to put gas into its shell and float off the sea floor. This probably looked a lot like that nautilus on the top right. Over evolutionary time, several groups of cephalopods lost their shells. That form is seen in the squids and the octopi. The octopus lacks a shell. The mantle cavity is carried like a bag behind its head. They have 8 arms, and in the center of their arms they have a mouth and a beak. They have a small radula. They also produce venom that enters wounds through glands in the mouth. Their eye is very complex and looks very similar to our eye, but it evolved independently from our eyes. This is actually an incredible example of convergent evolution, and we'll look at that a little more closely. The octopus propels itself using a siphon. This siphon pushes jets of water out. The octopus can also change the color of its skin rapidly. In this example borrowed from the primary literature, we can see that octopi can change the color and the texture of their skin. In this paper, researchers have tried to recreate that. This is an example of biomimicry. In technology, where we've taken examples from nature and tried to use them for our own applications. In this case, it would be handy to use as camouflage. Here you can see a comparison of the eye anatomy of a mollusk and a vertebrate. Remember that these eyes evolved independently. Any similarity between these eyes is a result of convergent evolution. Evolution converging on the simplest solution. You can see that both eyes feature a lens. They also both feature an iris. The placement of the nerve fibers is different. the nerve fibers occur behind the photoreceptor cells in the octopus, whereas in vertebrates, the nerve fibers occur in front of the photoreceptor cells. Lastly, the optic nerve separates the photoreceptor cells and creates a blind spot in the vertebrates. In the octopus, the nerve fibers run behind the photoreceptor cells, so they don't have to create a blind spot in order to pass through. On the bottom left is an octopus eye, and on the bottom right is the eye of a frog. Remember that these two examples of eyes have independent lineages and evolve separately.